So again, for those who don't know me, I'm Pete Denardis, the chair of the Board of Trustees, and it's my distinct honor to introduce Dr. Stephen Treon. Most, if not all of you, already know who he is. He's a luminary in the field of WM, and you can read more about him in the bio sheet, but I just wanted to state a couple things that sometimes, you kind of lose sight of just how important this person is to us. His accomplishment over the past couple of decades really speak for themselves, but consider in 1995, it was a very important year for the WM community. It's a year Dr. Treon received his certification from the American Board of Internal Medicine. And it's the same year that Dr. Arnie Smokler, a WM patient, began formulating what is now known as the IWMF. I can't stress how truly awesome and fortuitous it was that Stephen Treon chose as a newly minted doctor to hang his stethoscope and the future of his career as a researcher on focusing on such a rare disease as ours. And at the time, it didn't even have an effective treatment. He dove headfirst in the world of WM, so much so that he established a clinic devoted specifically to WM at Dana-Farber in 1999, the first of its kind, and has seen and continues to see thousands of WM patients from around the world on a regular basis, not to mention having published hundreds of articles and done groundbreaking research that has specifically led to the approval of treatments specifically for WM. I've been at meetings on countless occasions where, among his peers and fellow researchers, he has a calm, unassuming manner, but yet he gets right to the heart of the matter that's in question and submits astounding insights and encourages others to push the boundaries of WM research even further. He truly is a rock star among rock stars. Truly, many of us, I mean this honestly, would not be here today would it not be for the dedication that Dr. Treon has to our disease. So after his, I do want to mention one thing I really am glad to have the honor to present Dr. Treon, but I do want to mention that you do have the capability to give reviews of the evaluations of each of the presentations, and it's on the app. So once you go to the app, if you go to the home button on there and go to agenda, and then you just scroll down and click on each individual presentation and scroll to the bottom and you'll see, I know you can't see this, but you'll see presentation feedback. You click on that and then you can enter your feedback and your evaluation. Thank you. So after his talk, Dr. Treon will answer questions if there is time. And for our folks here, please use one of the aisle microphones again. And for the virtual audience, you can submit your questions by way of the Q&A section. So, Dr. Treon.
Good morning. It's such a pleasure, privilege, honor for me to be here. I have been coming to the IWMF forums now, I think, 26 years. I've got to say, thank you. And the best part about this is that you get the chance to see many friends and colleagues, but the better part of it is that we're all growing old together. And we're doing it quite well. So my task today was to talk about novel ways that we're targeting MYD88 and Waldenstroms. And I'm probably not telling you anything new that MYD88 or MYD88 is in fact public enemy number one when it comes to Waldenstroms, particularly the mutated form that we're going to be talking about. Before I actually give my presentation, I did want to have this in memoriam of my very close friend and colleague, Glenn Cantor, who I think all of you know was a member of the IWMF Board of Trustees. He was a doctor of veterinary medicine and also had a PhD and was a wonderful colleague. He was soft-spoken, but when he spoke, his words meant so much. And he counseled us on two very important things. One is the development of the drugs that we're going to talk about, the need to be able to come up with targeted therapies for Waldenstrom's and really move away from the era of chemotherapy to what we're going to talk about today, which is targeted therapies. He had many insights because he spent quite a bit of time in the pharmaceutical field developing drugs, and he was always happy to volunteer information, connect you to people, give you actually great insights into ways that we could develop these drugs. The other thing that I also want to say about Glenn was he was a champion for bringing in new blood to the research field for Waldenstrom's. We together had the honor of, along with our colleague and friend Tom Hoffman, who you all know, of chairing the Robert A. Kyle Career Development Award Committee. And, part of that task was to be able to come up with creative ways to attract more young blood to our field. He was a big champion of that. And at every meeting, the question that would come up is, okay, we have only 3 awards we can give in this cycle. How can we give more? How can we get more people interested? And the same for the Young Investigator Award that we give out at the International Workshop on Waldenstrom's. The more the merrier. The more funding we could get, we can bring in even more minds, to work on our disease. This is the reason why I am missing him at this meeting and missing his friendship and his collegiality. And fortuitously and strangely, as one day when he came with his wife, Inge Eriks, to my office, I found out that his wife's father was actually my chemistry professor when I was at BU. So you talk about the circle of life. We're all connected in many, many ways. Now, I want to say from the get-go, we have really moved into a new era for Waldenstrom's. In the old days, that Peter did a very nice job of sort of alluding the early days of Waldenstrom's and even my career. When we thought about Waldenstrom's, it really was a hand-me-down disease. We'd go to all these meetings, the CLL meetings, the lymphoma meetings, the myeloma meetings. We would look at all these great drugs that they were working on. And for us, it was a question of, will they give them to us so we can look at them in Waldenstrom's? Will they work? What kind of toxicities would they have? But it was all hand-me-down. We had to hope that whatever was being developed in all these other fields would work in our diseases. And we didn't even have clinical trials back then that were dedicated to Waldenstrom's. We were part of what we call basket trials, that, we'll throw in a couple of Waldenstrom's people along with everything else and see whether it sticks or doesn't. In the era that we spawned, and I think we were among the first, we now had dedicated trials to Waldenstrom's, but with hand-me-down medicines. And this is where the IWMF, I have to tell you, played a pivotal role. I go back to Arnie Smokler's time where he wanted us to be able to create the science in order to be able to create the new medicines. And that's what we're going to talk about today, to see how far we have gotten. So I want to show you also some pictures, again, the good old days. This is the second international workshop on Waldenstrom's. The first international workshop was actually held in Washington by Bruce Chesson. I don't have a picture for you. There were only 17 of us at that meeting, and the only thing we could agree to at that meeting was all the things we didn't know about Waldenstrom's. That's why there's no picture. But at the second, you can see, in fact, that we're starting to actually get a core group of people who are focusing on the disease. We all look great here, by the way. We're all much younger. Even Bob Kyle, you can see there, looks just fantastic. Stan Frankel is there in front. Thanos Demopoulos is there. Veronique Leblond. I mean, it was a really nice group of people, and we would start working together and coming up with new trials and new ideas for moving the field ahead. And very pivotal moment also was in 2003 when we had the second International Patient and Physician Summit. This actually met at UCLA in California, in Los Angeles. And this is really when we first started strategizing about understanding the basic cause of Waldenstrom's. What was that genetic, mistake that actually formed the Waldenstrom cell? And this was a powerful brain trust. You can see, of course, Zach Hunter, a very, very young Zach Hunter in the back. But Nikhil Munshi is in this picture, Andy Brannigan, Linda Pilarski, Olivier Tournelac, who actually does an incredible job now in France leading transplant efforts for Waldenstrom. But this was really the first brain trust to try to come up with how we were going to go about sequencing Waldenstrom cells and identifying the genetic mistake. Now, what we didn't have in those days was whole genome sequencing. And we didn't really have even a dedicated laboratory that could mount all this genetic innovation. And so in 2005, who I consider really one of the greatest heroes of our field, Peter Bing, who at the time was chairman of Stanford University, decided to make a personal investment in the dedication of a center focused on basic discovery for Waldenstrom's, and this occurred in 2005. And this really allowed us to start to pull together the talent that ultimately led to the advances that we're going to talk about. Then came whole genome sequencing. Finally, we had an art that could actually interrogate the entire cell. You heard about this from Zach Hunter. And this actually occurred in 2010. We had 30 individuals with Waldenstrom's for whom we got both bone marrow and also blood samples. And the technology was finally there, but it was super expensive. And not many people knew how to interrogate the data. We were among the first. I want to tell you, this is why you should all be feeling really good about the pioneering that the Waldenstrom's field was able to do, thanks in great part to the IWMF and to Peter Bing and many, many others. But we were able to actually use this art when there was only a handful of people who even know how to use the technology and even make the data sets. And what we're going to talk about is the mutation in MYD88, but I want you to really come away with how astonishing this technology is. We have in our cells over 3 billion DNA molecules that are strung together, and this is where we get all that coding for everything that the cell does. The mistake that leads to MYD88 involves only one out of those 3 billion DNA molecules. It gets flipped from one type of DNA to another molecule, and 1 out of 3 billion. And this is where whole genome sequencing played a pivotal role. And Zach Hunter should be credited along with the basic genomics group at at Dana-Farber for independently discovering this MYD88 mutation. And so it turned out that the discovery of MYD88 led to the best of ASH. That's the American Society of Hematology. It's the most prestigious hematology oncology meeting in the world. But that year in 2011, this was actually considered one of the most pivotal discoveries made in our field. And then a year later, the paper appeared in the New England Journal of Medicine. So finally, Waldenstrom's was actually leading the bunch. We were seeing innovation now finally coming our way and providing us really important insights. And whole genome sequencing taught us that 90% of the Waldenstrom patients had MYD88. I'll show you in a second, it's actually much higher. But the resolution for whole genome sequencing put it at 90%. Again, I want to credit both Peter Bing for providing the funding along with the IWMF and Judith May in particular. Judith really answered the call at this moment in time. She went out of her way to make sure she was excited about whole genome sequencing. She was seeing what potential it had, and she wanted to make sure that the funds got there so we could afford this. And it was very expensive to do this. Again, today, you can have your genome sequenced for about $1,000. In those days, it was really a very expensive endeavor. Now, specifically, the mutation that we find in most patients with Waldenstrom's is the L265P mutation. That's important for you to know. Because of that one DNA change, leucine. I think many of you may remember your basic biochemistry, but leucine's an amino acid, and it becomes proline at position 265. That's what this notation means. Proline is a strange amino acid because it contorts whatever protein it gets inserted to. It has really an impact on the structure. And that structural change is what causes MYD88 to go from a tame molecule to a very angry one. And both Zach Hunter and Leanne Zhu, who was in our laboratory at the time, were very, very instrumental in being able to identify and confirm the presence of this mutation. But Leanne actually went on and developed she was the first in the world to do this, to actually develop a PCR technology, which most of you in this room who have Waldenstrom's probably have been sequenced with her technology, with her ability, to detect this mutation. And now the detection rate is about 95 to 97% of all Waldenstrom's patients. Even people that have the prelude to Waldenstrom, what we call MGUS, or monoclonal gammopathy of unknown significance, we're able to pick it up in up to 80% of individuals. Now, this discovery and reporting in the New England Journal triggered many, many laboratories around the world. This is only a small handful, but using various methods, you can see that laboratories, big laboratories around the world, began to look at MYD88 and Waldenstrom's, and now there are over 50 confirmed studies. And the range for detection varies. It's probably around 93 to 97%. If you're using less sensitive technology like whole genome sequencing or whole exome sequencing, it might be a little lower. If you're using PCR, much higher. Now even a new technology called digital droplet technology may be boosting those numbers even more. But suffice to say that MYD88 probably explains the vast majority of Waldenstrom's. That small percentage of 3 to 5%, we've also sequenced those patients. Zach had a beautiful paper that appeared in Blood Advances a few years ago, and those are mutations that are, related to MYD88, but they're not MYD88. So with the discovery of the MYD88 mutation, a new era for this disease, commenced. I think very importantly, it helped with the diagnosis of Waldenstrom's. There are many diseases that actually make IgM, but by far Waldenstrom's has the highest rate of MYD88 mutation. This is a plot which shows actually based on density the individuals that have the strongest, detection of MYD88. And you might be confused because you're seeing that the MYD88 folks, let me see, I think this is a pointer, actually have what you might think is low levels. Well, this occurs because as the machine is looking for MYD88, if you have it, it starts detecting it very early. And if you don't have it, then it takes much longer. And in fact, anything above the gray line means that it's not being detected. So myeloma, you don't find it. CLL, very rarely you do find it. There's myeloma there. There's marginal zone lymphoma. This is a disease very much related to Waldenstrom's, but only a small smattering of patients have it. But you can see here how Waldenstrom's just screams at you. And so this was really an advance. But really where we started getting excited was trying to understand how MYD88 was able to cause the Waldenstrom cells to grow, and then, of course, work to find out how we could stop MYD88 signaling. This was actually part of the paper that we published in the New England Journal of Medicine, but also independent of us, a group at the NIH led by Lou Stout was doing the same work. And we both concluded that NF-kappa B, this is a very powerful growth signal in lymphoma cells, but NF-kappa B, we found, was actually being turned on by the mutated MYD88. We see it here. We use different technology to do this, but the way we did it was we actually used probes to look for NF-kappa B. And all these little lights that you see in here in these different screens represent NF-kappa B. But if we used an inhibitor of MYD88, just look at here how we turn the lights off. And that really was the technology that we were using to prove that NF-kappa B was very much affected. Now, when you look at these molecules, the thing to remember is it's like an electrical circuit. Things turn on things that then turn on other things. And the question is, what should we be targeting? So another hero, I think, for our field is Dr. Guang Yang, who spent really many, many years with us, almost 15 years with us. And this is the electrical diagram that he and his team came up with to explain why NF-kappa B was turned on. And it's a complicated circuit. It's in your handouts. I don't expect you to, there's no quiz at the end of this. But what I want you to do is come away appreciating how tedious it is to try to figure out how this relay race or circuitry actually works. And in the process, we learned a lot about this disease and came up with new drugs. So MYD88 is right here. Usually this is a good molecule. It's supposed to signal if an infection comes along. But when you have the mutation, the switch is turned on, and it stays turned on, and that's the problem. And so in this cascade is this central molecule called HCK. This is a very important molecule. We've actually deciphered exactly how mutated MYD88 turns on HCK. And we're going to talk a lot about HCK in a moment. But this is actually what turns on multiple different important circuits, including Bruton's tyrosine kinase. And you can see why we got so excited about knowing that BTK, Bruton's tyrosine kinase, was part of this relay race, because after all, it gave us an opportunity to develop all the BTK inhibitors that were starting to sprout. And this is why Waldenstrom's ended up becoming really a showplace for the rest of the world. And we received, because of this discovery, tying together the genomics with how BTK was turned on, we received the first ever breakthrough designation for any oncology indication for fast-track approval by the FDA. This was a newly established way to move drugs forward. And Waldenstrom's was that first paradigm for using this pathway. And of course, this led then to the first drug approval for Waldenstrom's, that being the drug ibrutinib, the BTK inhibitor. This was, as you can see, a lot of happy faces. We had just met with the FDA in June of 2014, and they were just thrilled. They were over the moon by the nice data set that we brought in, not only the preclinical data, but also the clinical study that Leah Palumba, myself, and Ranjan Advani had run. And so since that time, there have been a lot of trials looking at BTK inhibitors in Waldenstrom's. I don't expect you to memorize this slide either, because I've actually condensed it for you. And I just want you to come away with the snapshot. I don't care which BTK inhibitor we're looking at. I don't even care whether the patients got it in the front line or they got it, after being treated. The data is very consistent that about 90% of all patients will get at least a minor response. 80% will get a major response. Even some patients will get over 90% reduction in their disease burden with just one single BTK inhibitor. And on average, 75% of patients will have their disease remain in check at 4 years' time. Now think about this. In contrast to the previous, therapies that we were using, which might last a year or two or three or maybe longer. But I also want you to see why we're going to talk about new drugs and why we need them. And that is that despite all this very exciting data for which we should be very proud of, it's really impacted the survival of Waldenstrom's patients, we see very few or no complete responses. So if we are to dream about a cure, and I know in Pete's mind this is the case every day, we've heard Carl Harrington talk about the road to a cure, but if we are going to be on that road to a cure, we need more complete responses. And so this is the reason why we've been trying to understand, what it is that's still keeping these Waldenstrom cells alive, even though we're able to sort of, suppress their growth. Now, one potential explanation that we looked at was the CXCR4 mutation. This was also discovered in our laboratory thanks to whole genome sequencing. Zach should take a bow on this completely because it was his informatical skills, computational mathematical skills, that allowed him to re-engineer the algorithms that we were using for whole genome sequencing. And then one day he comes to us and says, you know what, I think I found another very important mutation, and that involves CXCR4. Now, CXCR4 is an interesting mutation because this actually is a receptor, and many of our cells have these antennas that are always looking for signals to respond. And this CXCR4 molecule responds to this other protein, CXCL12, that's made by the bone marrow. This is one of the reasons why Waldenstrom cells like to go to the bone marrow and hunker down. About 40% of all Waldenstrom patients have this mutation. We also reported that it was a plenary paper in the journal Blood. And then thanks to Dr. Yang Kao in our group and others, we were able to decipher exactly what it was doing. And what it was doing was really turning on other very powerful molecules that allowed the cells to grow and survive, but also home towards the bone marrow stroma. And when you have the CXCR4 mutation, these are patients that usually have very high IgM levels, might have hyperviscosity syndrome, but also through our modeling, as well as the modeling that was done in Dr. Irene Gobriel's lab, we learned that they play a role in drug resistance. So we began to say, well, maybe CXCR4 is part of the reason why we're not getting to those complete responses. So let's try to dissect out all these trials and look at the impact of CXCR4. Again, please don't feel obligated to memorize the slide. I've summarized it for you. And the long shot is, if you have the mutation, it does impact outcomes to BTK inhibitors. It takes longer for somebody to respond. You don't get as deep a remission as possible. The ability to keep the disease in check is a little bit lower. You actually see the progression-free survival is 59% versus 75% at 4 years, so this is a difference. But it doesn't explain why we're not getting those CRs, because even when people don't have the CXCR4 mutation, we're still not seeing complete remissions. So there has to be more to that story. And so I do want to tell you, one of the important studies that has helped us give us some insights has been the Aspen study. You're going to hear about this by other people, but this was a first of its kind, comparing one BTK inhibitor against another, so zanubrutinib versus ibrutinib. And we did actually look at the impact of these different drugs, again, on CR rates. And you'll see, in fact, that in the Aspen trial, both of these BTK inhibitors, zanubrutinib and ibrutinib, did well. I mean, major responses were about the same, 80%, slightly more with zanubrutinib, so maybe there's something more to the zanubrutinib. But again, very few CRs, in fact, none that were reported in this trial. Even among people that didn't have the MYD88 mutation, I think there may have been one CR in total. So again, this was not the answer, that if you used one BTK inhibitor versus the other, that you were going to get complete remissions. But zanubrutinib did surprise us. We did see among the CXCR4 mutated patients deeper responses, even better overall response, suppression of the disease over time. So there were some differences that actually played into having the CXCR4 mutation. So what was keeping these Waldenstrom cells alive? And this, I want to acquaint you with two very important terms. One is intrinsic resistance. This is something that the cell already has that's making it completely, that makes it, though vulnerable to BTK inhibitors, doesn't allow you to wipe out all these cells. So that's intrinsic. And so I want to go back to that very complicated electrical diagram, because this is where we start getting our answers. And I mentioned to you this pathway of MYD88 turning on HCK that then turns on all these other molecules. But look here, it also turns on BTK, NF-kappa B, right? But there's also a second pathway here. This is a bypass route mediated by these molecules called the Iraq molecules. There's two of them, Iraq4 and Iraq1. They also turn on NF-kappa B. So even if you're poisoning or shutting down BTK, here's the bypass loop here that's also triggering NF-kappa B. Now, you may say to me, how do you know this? Well, I'll tell you. It's because when we were treating patients on our ibrutinib trial, we were taking their cells and we were looking to see which one of these molecules was still on. And in fact, these were just a handful of patients who were treated for 6 months on ibrutinib, and we could shut down BTK, but the Iraq molecules were still on. And then when we took those cells and treated them with both Iraq inhibitors and ibrutinib, we could start killing them. So we already had data that was telling us that these Iraq molecules were in fact very significant. And more to this study, we then started knocking down in Waldenstrom cells both the Iraq-1 and Iraq-4. Knockdowns mean genetically, we can turn them off. And I'm going to teach you a little bit about protein chemistry. So here's the vectors. We actually put into the cells these vectors that actually turn off genes. So here is Iraq-4, here is Iraq-1, and this was very elegant work done by Dr. Shalu in our laboratory. These are Waldenstrom cells, and this is the protein Iraq-4 in its native state because we use a viral vector here that doesn't affect it. But when you do the correct, knockdown, you can see how much more pale these are. This means that we're actually toning down the Iraq expression. And what we found here was that we got more cell death. These are readouts for cell death if we hit Iraq-1 versus Iraq-4, but both actually contributed to the survival of these Waldenstrom cells. Armed with this information, we then put together the dream team. This was a medicinal chemistry dream team available to us at Harvard. Dr. Nathaniel Gray at the time led the effort. He's now at Stanford. Dr. Sarah Berlaghi is still at Dana-Farber and Harvard. She's part of our ongoing team. Dr. John Hatcher, actually, we recruited to the Bing Center, so he's now on staff. And thanks to the generosity, I'm not allowed to actually share the name of the family, but thanks to the generosity of this amazing family, we have now a investigator chair endowed for his ongoing, retention and support. And Dr. Jinhua Wang is also part of the medicinal chemistry group at Harvard. These are my heroes because these are the individuals that are working day and night to come up with targeted drugs for Waldenstrom. Now, we're not going to get into all the fancy medicinal chemistry today. You have actually a handout. But their efforts actually succeeded in making us an Iraq-1 inhibitor. It was the first of its kind. No one had made an Iraq-1 inhibitor at that point in time. And what I do want to teach you, though, is that when you're doing all this medicinal chemistry synthesis, usually some of the molecules will have other targets, not just Iraq-1, but look at all these targets. We call these dirty molecules, okay? But you can see here that as we try to innovate and come up with multi-generational improvements, we ended up with this Iraq inhibitor. And you can see, in fact, how clean it is. If I were to put on here zanubrutinib or ibrutinib, or even pirtobrutinib, you'd see a lot more of these blotches. The fact that we got such a clean molecule excited us. Now, when we combine this molecule, which we call JHX11901, by the way, I only learned like a month ago. I just thought, okay, this must be some, designation that's universal. No, this actually is John Hatcher. Each one of these medicinal chemists name these molecules after themselves. And it was during one of our conference calls that it just dawned on me, because I could see then Sarah Berlaghi, SB, her molecules are all the SB series. Jinhua Wang, JW series. I caught on at that point. They're a lot more vain than we are in the oncology world, but I love them. But what excited us was when you took that Iraq-1 inhibitor and combined it with ibrutinib, and these are again MYD88 mutated cells, all these red blotches that you see, because we use different doses for each drug, represent synergy. Not just additive killing, but synergy, that we're actually, by combining Iraq inhibitors with ibrutinib, we were able to synergistically kill these cells. So that meant we were achieving our results. Now, we then did what we call pharmacokinetic studies, where we give mice or rats the drug, usually intravenously and also by mouth, to see what sort of characteristics of distribution are. And so we can get, if we give the drug intravenously, we can get very high levels. These are micromolars, well above, like 100-fold, well above what we need to shut down Iraq. But when you give it by mouth, it's a different story. We only get about 1% or less absorption. It's too low. That's a problem if you want to give the drug orally. The numbers that you want to see are in the 30 to 60% range. And Glenn was really wonderful at this because he could look at all this PK data and tell you this is what we need to do. Now, why I'm showing you this is because I really want you to understand how much work goes into this. It takes a while. And sometimes you improve on the targeting of a molecule, but then you lose the pharmacokinetic ability. Sometimes you advance the pharmacokinetics, and then the drug doesn't work as well. There's lots of reiterations, and that's why that medicinal chemistry dream team is so important to us. So they have been working, and to our benefit, they've been able to actually make still relatively selective molecules. In fact, they've gone one step further. Not only now targeting Iraq-1, but also Iraq-4 with these medicinal chemistry improvements. We now have a new generation of drugs that we're working with, but we're starting to see increased what we call bioavailability, increased absorption. So about 15% of the drug now is getting absorbed. That actually could make it into a clinical trial. It's on the low side, but you can actually do it. But we're still innovating on this. So I want to now introduce you. Oh, darn, I wasn't supposed to show you that slide first. But I want to tell you now what the IWMF has funded, which I think is very exciting. Sometimes these molecules, these signaling molecules, not only turn on other pathways because they basically hug other molecules and charge them, but also they play a very important role, what we call scaffold role, where they can anchor other molecules, and that allows them to be able to propagate growth and survival signals. So why not try to destroy the molecule altogether instead of just shutting it off? And this technology was also invented at Dana-Farber called the PROTAC technology. Basically what you're doing here is if you see a molecule that you don't like, you can tag it, target it, and drag it to the wastebasket that every cell has. Every cell has an ability to destroy molecules and recycle them. And this PROTAC technology is reminiscent. I'm sure all of us are probably in a similar age group. I apologize if you're not. But do you remember playing Pac-Man? Right? I mean, that was the thing I did, when I was in college. Those little breaks that we had, you'd have to show up with your quarters. And we played Pac-Man. This is what we're doing. We're playing Pac-Man. And we're going after the molecules that we don't like. And Iraq-1 and Iraq-4 are on that list. And so thanks again to the wonderful work of John Hatcher. Again, pay attention. These things are called JH. We've succeeded in making Pac-Man against these Iraq molecules. And again, I don't have to show you now. Now you know all about this. This is what these proteins look like. And then you apply these drugs. There's different series at different doses, and you can see how we're eliminating them or we're dimming them down. We're very excited about these Iraq PROTACs. And in fact, we've made a PROTAC here called the John Hatcher X1305-1, which actually chews down both Iraq-1 and Iraq-4. And these are very, very potent drugs against MYD88 cell lines, not only Waldenstrom's, but MYD88 mutations exist in aggressive lymphomas, lymphomas that go to the brain, other types of B-cell malignancies. So we're now seeing activity for these molecules even in these other cancers. So again, we're leading the way, and thanks to the IWMF, your support, their support is translating into benefits for other patients affected with these cancers. So I now want to talk about acquired resistance. You learned about intrinsic resistance when you've got something in the pathway that's not allowing you to kill off a cell. Acquired resistance is when you're responding to, let's say, ibrutinib or zanubrutinib, and then you stop responding. You become resistant to it. That's when we use the word acquired. So the most common way that patients develop acquired resistance, and this was again identified in our laboratory, it had been previously shown in CLL, involves a mutation at the site where drugs like ibrutinib bind to. Ibrutinib is considered a covalent BTK inhibitor. That means when it latches on tightly. And whether you're thinking about ibrutinib or zanubrutinib or acalabrutinib, these are all covalent BTK inhibitors that all latch on to this amino acid called cysteine at position 481 on the BTK molecule. This is where it latches on and it binds. But if you develop a mutation, then the ibrutinib no longer can bind there, and that's how the cell now is able to grow and survive. Now, again, I want to show you why I'm excited about this, because we've learned a lot. This is, again, Lian Zhu, who identified these mutations. She actually had a very interesting note when she did this. Not only were patients who were progressing on Waldenstrom's developing one type of BTK mutation, in fact, some people had 4 different ones. So it means that these clones likely already exist in patients, but when you're putting the drug on these patients, ibrutinib, the cells that don't have the BTK mutation are dying off, but then you have all these weeds growing in their place that carry the mutation. So again, this is an acquired mutation. And Lian did all this work. And I'm going to show you one patient here just to sort of show you how fast the weeds can crop up. This is somebody who, at baseline, meaning before they got treated, we couldn't detect any of these mutations. So they have to be truly microscopic because our DNA interrogation didn't pick it up. By month 11 that the patient is on, again, we can't pick up any of these BTK mutations. By month 22, we're starting to actually see 2 different ones. And look at the percentage, 0.71% for one, 0.19%. So still tiny little critters, in this among all the clones. But by month 35, you can see now they're occupying, somewhere around 30% of the clone. But you see 3 different mutations that have cropped up. And these cells are becoming resistant to ibrutinib. So in my laboratory, I was blessed to have the mother and son team, Jaji and Shah, who began to model these mutations. Again, a first in the entire field of BTK inhibitors. This was also a plenary paper in Blood. And what they found was that when you had one of these mutations in BTK, this pathway got turned on called the ERK pathway, ERK. This is also a very powerful signaling molecule in Waldenstrom cells, but what it does is it flushes out inflammatory cytokines, proteins, that then sort of inflame out the bone marrow stroma. And they also do something else that's very interesting. Even the cells that don't have the BTK mutations, these cytokines teach them or trigger them to become resistant also to ibrutinib. So they start talking with each other. And we did this by doing these very elaborate studies called co-culture experiments. There's just a membrane here. These cells don't actually touch each other. But if you have a cell that doesn't have the BTK mutation on each side and you add ibrutinib, they all die. That's exactly what we expect. But if you put on one side of the membrane a BTK mutated cell and on the other side the ones that don't have it, and you add ibrutinib, guess what? They both survive. And if you can figure out what cytokine is actually doing this, that's being released by these cells, and you block it, you can start killing these cells again. And so they talk to each other. The bad apples are telling the good apples, don't react to ibrutinib. And so this is called paracrine means of inducing resistance. We were the first to actually show this. But it's an incredible concept to think how cells are talking to each other and teaching them to be bad. And this is why shutting down pathways like, the ERK pathway may be a way forward for us to do this. And this kind of shows you once again how ERK figures into this picture. Here's NF-kappa B, which promotes growth and survival. Here's ERK that flames out with all these cytokines that then teach other cells not to respond to ibrutinib. And now we have actually some new drugs that can help us. One of these drugs is pirtobrutinib, and I know you're going to be hearing about this drug. This is a non-covalent BTK inhibitor. It binds at a different site on BTK, not the one that becomes mutated, but a different site. And what we were able to show, with the work that we've done is that you can actually shut down the ERK pathway. Oops, I don't have time today to get into all the details, but you can shut down, here we go. You can shut down that ERK pathway by using a drug like pirtobrutinib. We just submitted this paper. It's been presented before at the American Society of Hematology. The other way that we hope to shut down acquired resistance is actually to go one step above. So instead of, trying to hit BTK, maybe we should be taking out HCK. Because you can see here, HCK does a lot of things. It supports BTK and NF-kappa B, but it also turns on ERK as well as other survival pathways. And so the IWMF is actually funding us to be able to come up with drugs that can actually poison HCK. And I want to show you some of these very exciting developments that we've done. And I'm going to move a little bit further ahead. I showed you the dream team. We already appreciate them. But we've made now very powerful HCK inhibitors. One of the lead drugs here is called KIN8194. It took us over 400 analogs of synthesis and triaging to be able to come up with this drug. This was also published in the very prestigious journal Blood. And you can see here that it hits HCK, it even hits BTK at very, very low levels. These are what we call sub-nanomolar levels, so very, very potent inhibitor. And we showed in these experiments that have been published that you can turn off HCK and BTK. I won't bore you with these details, but I do want to show you how selective these drugs can be against MYD88 mutated cells. And again, we used Waldenstrom cells for these experiments, as well as aggressive lymphoma ones that had the MYD88 mutation. And you'll see how they selectively kill MYD88 mutated cells versus cells that don't have the MYD88 mutation. And more importantly, here you can see that the Waldenstrom cells from patients were being killed. This is the kill rate, whereas their normal immune cells, T cells, B cells, were not affected. So this specifically shows us that if we can hit HCK, we can target very selectively MYD88 mutated Waldenstroms. I actually love showing you these experiments. These are experiments where mice were engrafted with a very aggressive MYD88 tumor, the TMD8. These were tumors growing on the flanks of these animals. The animals were treated for a period of 12 weeks and then stopped. Many of them were cured. This is something that we never see with all these drug studies, to actually see curative potential. Now, if you had the mutation in BTK, inserted, it's a different story. You can actually retard the tumor much better than, and it was very much dose dependent, but we're not seeing the cure. And what this really means for us is that if we're going to develop these drugs, we should be getting them into patients earlier, not later, if we really are looking for a cure. Now, I want you to feel good. Again, IWMF and all of us here who support the IWMF, the drug actually is active in other lymphomas, even those that don't have the MYD88 mutation. This was a paper just published by the Lantermans group at the University of Amsterdam showing profound activity for this drug in mantle cell lymphoma. It has a different type of signaling cascade than MYD88 mutated Waldenstroms, but it can be overlapped with KIN8194. Mantle cell lymphoma is one of the worst lymphomas that you can have, and this is why seeing our ability to generate data with these drugs that will affect individuals with other cancers is very humbling. So we're very excited. Now, let's go on and talk about John Hatcher. John has been working to actually turn these HCK inhibitors into PROTACs, into degraders. we have been very successful in actually manufacturing 2 different ones. And again, this is really a big breakthrough. Again, look at the amount of protein for HCK in a cell, BTK, and then look and see what these degraders are doing. You know, in the case of this one here called DFCI-006, complete degradation of HCK, you can't even find it. And you can see how BTK, which is so strongly expressed in these cells, is being degraded. And these are just 2 different doses that we use, 0.1 micromolar and 0.5. The second degrader, called DFCI-05 here, we can also degrade it. Not as much as we did with the 06 compound, but you can see the degradation also extends to BTK. And overall, these are very clean and well-tolerated molecules. This is where I get excited, because even when those tumors were growing on the animals and we gave these drugs orally, we could once again degrade very nicely HCK. Again, look at how much there was in the animals that did not get the drug. We start seeing a decrease based on increasing dose. By 30 mg per kg, we're seeing much less of it and no BTK at all. So this shows us that we're able to achieve this even in the animals giving the drug by mouth. And more importantly, in 5 days, look at here the tumor growth. This is what happens to the tumor if there's no drug. If you're using these inhibitors, the PROTAC-05, you can actually, 06 I should say, you can actually completely suppress and degrade the tumor itself. So we're very excited about advancing these molecules. This is preliminary data, by the way. I just want you to know that when I come to these meetings, Pete is expecting me to show something exciting and new. and this is why you're seeing exciting and new data that's really hot off the presses. Now, let me give you some more good news. You give this drug by mouth, you already can see the data, you know, that it is getting to the tumor. We're getting about 40% of the drug, you know, being absorbed, and we're getting very, very high levels of the drug in circulation. So these are going to be winners in terms of being able to move them forward as oral drugs. We're also looking at repurposing drugs. Dr. Shana Sorosiak, who's one of our clinicians, along with Dr. Sharong Liu, who is the Michael and Elizabeth Bell Fellow. Mike and Elizabeth endowed his fellowship. But there is an interesting drug. It's approved by the FDA for the treatment of myelofibrosis, except it does things that we like, which is it's able to shut down HCK and EROC. It's a dual inhibitor. And this we were able to very nicely show and report at the American Society of Hematology can actually target Waldenstrom's and ABC-DLBCL cells with the MYD88 mutation. And we're moving ahead to opening up a clinical trial. So we have the ability to even repurpose drugs other than those that we discover. So in concluding, I want to tell you this is all a team approach. We're a big family, much bigger than we were when we first met in Washington at that first fateful meeting. And what I am really excited about when I think about the future of this disease is all the young talent that we now have been able to bring into this field. Your support of the IWMF is critical for us to be able to fund these incredible minds that have focused their lives, you know, on our disease. And I just want to acknowledge both the IWMF, the WMFC, the Waldenstrom's Macroglobulinemia Foundation of Canada, the Leukemia and Lymphoma Society, the NIH, as well as Dana-Farber, and then you can see all the wonderful foundations and funds that have been set up to help support this work. And with that, I will stop and happy to take any questions if Pete allows me, but I'm at your disposal. Thank you very much for your attention.
Okay, so yeah, we do have a couple minutes, so please come up to the microphone if you have questions. And while we're, okay.
So I presume with these candidate molecules, you establish an IP position and that you talk to pharmaceutical companies about really funding full-scale clinical trials. Could you talk a little bit about that process?
You know, that's a wonderful question because, you know, there is this so-called valley of death where you can develop exciting molecules, even get astonishing papers in top journals, and then what happens, before it gets to be a drug? Well, many things have to happen. You have to raise the money to be able to do the advanced toxicology studies, pharmacokinetic studies that are needed. You need to raise money to be able to pay for the clinical grade of drug. These are all manufactured at a grade that's doesn't have the superiority in terms of purity. And that's a very expensive process. I have to say, just to be able to scale up drug for these animal studies could be as much as $1 million. So we're talking real money. And that's on top of being able to then do the IND enabling studies that the FDA needs to see in order to be able to say, okay, clear for human trials. So these are very expensive propositions. Your insights on IP are very important. For those of you who may not, be familiar with the term, it's basically whether you've got patents on these drugs. And patents come in different ways. You can patent the composition of the drug itself, and you can also patent the usage. They're both critical to have those types of patents, because when pharma's thinking about investing in developing your drug, they need to know that they have protections. And if you don't, then you're in the valley of death once again, because, who's going to come up with the money to do this? And it is sad, because there are a lot of drugs that are in the valley of death that are fantastic drugs, but because there is no IP, intellectual property positions, no one wants to pick them up. And picking up, these drugs is important. There are many drugs out there that we and others have developed over the years that sit in that valley. But being able to move these drugs forward becomes a problem unless you have this. Now, the nice thing is, I think for these drugs, most of them we have IP, and I think that will help actually move these drugs ahead. But that's a very good question, because I really want people to understand what it takes to be able to move from a concept to actually having a drug. Please.
I have a question, but let me first say, thank you for bringing up Glenn Cantor, and he was a member of our support group, the Oregon Southwest Washington support group, because he moved to Bend, Oregon, and was a valued member and was really approachable. I had an iron deficiency problem, and he had IV iron, and he answered all my questions. And we miss him terribly. And I don't think you mentioned this, but I think people should know it wasn't Waldenstrom's related. Right. His death, it was a tragic snowmobile accident in Alaska. So thank you for talking about Glenn Cantor. My question is, I didn't catch the name of the IRAC inhibitor, but it was interesting. The pacritinib is an HCK and an IRAC inhibitor, and has a name already. So how far away are we from actually using these drugs? I assume you're doing clinical trials on the drugs. And when do you foresee using these combinations?
No, those are all good questions. So pacritinib already is FDA approved. And so we're looking at repurposing that drug for Waldenstrom's. And we actually have the green light to move ahead with the trial. It will be, funded by the company. Dr. Sorosiak will actually be leading that effort. The other drugs still need to be, vetted out. They need the IND enabling studies. But I'll tell you, we've made so much progress in being able to, advance these drugs. I suspect by year's end, we're going to have a lot of the toxicology work and pharmacokinetic work done. And then it's, again, coming back to what you mentioned, which is courting the pharmaceutical companies. And we have really a good rapport. I'll tell you, with the development of ibrutinib and zanubrutinib and acalabrutinib and pirtobrutinib, we've really positioned ourselves in a wonderful way with pharmaceutical partners that I think are very interested in helping us to keep moving these drugs along. And thank you for mentioning, the cause of death for Glenn. This was a completely unrelated, unexpected death, and truly sad. Yeah.
We spoke of this briefly outside, but you mentioned it again here. So WM has often been associated with a counting procedure. What's your MGUS? You're not getting anything until your MGUS goes over a certain level. And you mentioned that perhaps earlier treatments might work better instead of treating the symptoms, perhaps go after the causes. Would you talk a little bit more about that and what you mean by that and
Yeah,
its
so
consequences?
I won't get into a specific, patient case because you're obviously.
We're not talking about me, it's okay.
Well, that's what I was trying to say. Because it's really hard without a lot of information. I think the point that I was trying to make earlier was that, we now have a lot of good drugs, and sometimes patients are resistant to being treated. What we don't want as physicians to have the patient fall off the cliff before you lend a helping hand. The way we designed our consensus panels was to formulate policy around and recommendations around when it's appropriate to treat. And there's actually check boxes, which I'm sure somebody will be covering here. But bottom line is somebody is symptomatic, we tend to treat. We do follow the IgM. We don't necessarily treat the IgM. But when you're above 6,000 with the IgM, we do worry a lot. And in our practice, we will treat because the risk of hyperviscosity causing something bad is way too high to just sit on it. I've seen it. I've seen people lose their vision or have strokes, heart attacks, because they said, hey, I don't really care. I feel good. And that IgM was way too high. I don't want you to do that. And that's why, again, when you look at somebody, instead of putting a Band-Aid on it, sometimes you need to treat the disease. And that's the point that I think I was trying to make.
Yeah, well, and you mentioned it here, and I was wondering if there were any other, were there any rules coming up upon, rules or procedures coming up upon that?
Well, I just want to say for the audience, I think last year I was able to cover that at the international workshop on Waldenstroms, we had 7 consensus panels that radically, updated much of our policies and recommendations around treatment of the disease. But these will continue to happen because as we get newer drugs and get newer information, we're going to need to continue. And I think this year we have at least 3 updates planned and different segments of care for Waldenstroms. Peter?
Okay, thank you. I think we're just have maybe 1 more minute left here. I want to have a question from the people in the audience and virtually. So if you could just answer it quickly, if that's possible, and then we'll be done. But the question is, how do you define complete remission? So if someone's IgM is lowering and goes down to the normal range, is that complete remission?
No, not necessarily. So you need a normal IgM. The monoclonal protein that's, the IgM needs to disappear. You need to do a bone marrow biopsy that shows, no evidence of disease. If the patient had enlarged lymph nodes or spleen, those have to be normalized. Those are in essence, the sort of criteria for achieving a complete remission. We have not yet entered the world of molecular remissions. I really hope as we advance our therapies we will, and that's where we use MYD88 to tell us, do you have any disease left in your bone marrow? We have the perfect tool to assess molecular complete remissions. We just need better drugs and better ability to get more people into complete remissions.
Okay, thank you. Thank you, Dr. Treon, for an amazing presentation. It's exciting to see the new agents that are coming down the pike, so I really appreciate the work that you folks are doing.
And you should all give yourselves a round of applause because it's really the IWMF that has driven much of this progress, so thank you, Peter.